second to last one for the semester here. So we're very excited to have Dan Larremore here. He's an assistant professor in computer science at CU Boulder and also works at the Biofrontiers Institute, but he's really a mathematician. And applied mathematician, you feel like that, and that counts. And he also works a lot in computational biology, and you're going to hear an example of that on complex networks and malaria. So please take it away, Dan.
Thank you. Thanks for having me. I grew up in Wheat Ridge. I went to high school in Lakewood, but I've never been on the Mines campus. So anyway, it's really a pleasure to be here. I'll tell you about some of the work that we've been doing in my group where we're trying to use methods from complex networks, statistical inference, and modeling to try and understand more about malaria parasites, how they evolved and how they spread, and how math can actually really on the ground help us have better estimates of the things we need to start decreasing the number of people who get infected with malaria each year and die. This is a map of the problem. It's a really sad map, actually. This is prevalence of Plasmodium falciparum, so that's the parasite that causes the most virulent form of malaria, among 2 to 10-year-olds. And so the darkest parts of this map are places where over 40% of the children, 2 to 10, have parasites in their blood. You can see that even outside of this range, you have prevalence across South America, Sub-Saharan Africa, as well as Southeast Asia. And the hardest part about malaria is that it's hard to access. It lives outside the public health infrastructure. So some of the work that I was doing in Kenya, this was the neighborhood just outside of town. And if it just looks like sort of trees with scattered buildings, that's what it is. But even in the concentrated areas, the healthcare services are a little bit tricky. So this is a photo I took of the orthopedics department at the district hospital. These are areas where there are not enough resources and where the funding for health in general is not really enough. The reason that I took this photo instead of others is that there weren't any people in the photo, and I didn't want to, take photos of people without their consent here. So this here is a list of prices for services on the wall. So these are really places that are challenging to get resources to. But the other challenge is not just that malaria lives outside the public health infrastructure, but where it lives in the body. So it lives inside red blood cells. So these are the same kind of stain, but at different levels of magnification. And if you can see this little blob with a ring attached to it, these different parasites in different stages, that's where the parasite lives. It spends most of its time inside red blood cells, hiding out. So what we'll talk about today are various efforts to use the signals that we do have that the parasite expresses on the surface of the red blood cell to figure out what it's up to while it's hiding out. So in a nutshell, my work is mostly on mathematics and methodology to understand malaria's immune evasion genes at the scales of both individual parasites and at the scale of populations. And today we'll talk about some network methods to decode their evolution, some inference methods to rigorously compare 2 parasites when the underlying data and our sampling is really flawed. And then just an example of how we can apply these methods in the context of epidemiology in a low transmission setting in Western Senegal. So, to dive right in, the malaria parasite has a life cycle that's in both the humans and in the mosquitoes. We're just going to arbitrarily cut that cycle and say that it begins when a mosquito bite introduces parasites that go into your liver and there they replicate. A little bit later, the parasites emerge en masse, and they enter this box here with the red boundary, and that's what's called the blood stage. That's where the parasite spends most of its time. So a parasite will find a home inside an individual red blood cell, and over the next 48 hours, it replicates. And at the end of the 48 hours, all the parasites, in a synchronized way, blow up their red blood cell homes, and now all of those parasites find new homes inside new red blood cells. And this synchronized process repeats every 48 hours. So if you're interested in the dynamics of synchronization, there's some really interesting problems here that I think are yet to be cracked open. This process continues and continues until eventually some of those parasites form a sexual stage, they make their way near the skin, and then a mosquito bite takes the parasites into the mosquito side. And then there's a whole other side that we won't talk about that takes place inside the mosquito. The key here is this blood stage. What happens is the parasite remodels its home. So this is a false color image of 2 healthy red blood cells. It's a nice donut shape. And an infected red blood cell. You can see that it's contorted. It's actually changed its conformation. what's happening here is, if you can see a little bit of texture on the outside, the parasite inside there is manufacturing a protein, which it exports to the surface of the red blood cell that acts like a Velcro. And that allows the parasite to bind to endothelial cells, which are all throughout the body. So in this photo, what you can see is an endothelial cell in the background here, and these are infected red blood cells that are sticking to them. Okay, so why does the parasite do this? It's a consequence of an evolutionary arms race. So the parasite really takes a nice healthy red blood cell and turns it into this mess for a good reason. The reason is that the spleen mechanically filters blood. And as the parasite's replicating, it stiffens the red blood cell. So if it were to freely circulate, it would just get filtered out easily by the spleen, and now you don't have malaria. So the parasite avoids circulation by using those sticky surface proteins. This is a process that we call sequestration. But the immune system is smart. It targets those red blood cells because it sees this foreign protein on the surface. So what does the parasite do? It expresses a different camouflage pattern on those surface proteins, and this is something that we would call immune evasion through antigenic variation. And an antigen is just something that the immune system can identify and react to. So the parasite uses one strategy, the immune system catches up. The parasite uses another strategy, the immune system catches up. And so on, and so on, and so on. So the parasite is sequentially evading immunity. So over time, the immune system has these nice memory cells, and so immunity to the various tricks that the parasite has grows. So the parasite makes it worse. They shuffle their VAR genes, and these are the genes responsible for creating that protein, making new camouflage patterns. This is something that we call diversifying selection through genetic recombination. And the mechanism here is really important. The parasite takes two different versions of the gene, and through crossover events, almost like a record needle skipping between the two, it reads a little bit from one, a little bit from another, so the next generation has this mixture. So, the problem for us is that the very same processes that enable the parasite to survive, this diversifying selection by genetic recombination, also disable our standard ways of analyzing these kinds of genes. So, here's a little bit more background. Each parasite has 60 different VAR genes. So I kind of think about this as about a deck of cards. The parasite has about one deck of cards worth of strategies to deploy. So I'm just representing these as lines here, color for each one of these different cards. But actually each VAR gene is composed of a variable number of different domain classes. These are just statistical descriptions of the gene content. And so these are the colored blocks that you see here. But it turns out the first domain is usually of the same class. It's a little bit helpful. So that's what this blue-gray box here is. Still, it's the same class, but there's widely different content. And that's because the parasite has been mixing and shuffling these since before humans were even a species, when the parasite infected our ape ancestors. So for instance, if I have one version of this and another version, I'm just representing differences in them by different patterns. So you basically have 60 different versions of this first domain here. So here's a little bit more about the mechanism in a very cartoon form. These VAR genes evolve by recombination. So if in one generation you have 1 and 2, in the next generation you can have what we call a single crossover event, or you could have multiple crossover events where you end up with this sort of mosaic structure. What's worse is that this doesn't just happen in the blue domain here, it can happen in the other domains. And so you can see that, yes, while you can have crossover events in these different domains, you may end up with progeny that have entirely different combinations of those domains. So what we have is a highly unstable gene family, meaning that from generation to generation they fluctuate a lot. They're mostly subtelomeric, so they occur out on the unstable ends of the parasite's chromosomes. They vary a lot in length, so this is 3.5 to 7 kilobases, because you can have a lot of fluctuation in the length of these genes. And there's so much variability that what people typically talk about in terms of the gene was 95% conserved or 80% conserved, the numerator and denominator there don't even make sense to compute with. So they're weird.
How is it that the recombination always will express a protein that can be suitable for camouflage?
So why is it that these recombinant proteins actually work?
Yeah.
It turns out that the parasite has disabled one of the standard DNA repair mechanisms, and it only uses a template-based mechanism. So in humans, if we have some kind of problem, there's like a double-strand break, things will get repaired using something that will be in frame or out of frame that might mess up the actual protein. What happens in the case of these genes is because that pathway doesn't even exist in the parasites, they always recover from a template, which means that when they see some break, they kind of look elsewhere in the genome and try and find something else to read from, and they do an exact match. And so it always ends up as an in-frame new gene. Yeah. It's an interesting result of this disabled mechanism. So really the problem is that our standard methods can't handle these genes. So phylogenetic trees are really the workhorse of a lot of genetic analyses. But they assume evolution by mutation, insertion, and deletion, which are bifurcating processes. So what I mean is, if you imagine that you have some set of genes and they're all identical, clonal, and then a mutation arises. You can model that as a split, right? You have the genes that don't have the mutation, you have the genes that have that mutation. Now if you have another mutation or insertion and deletion in one of the subpopulations, that's another split. And so the data naturally conform to a tree. Whereas what we have here is this mixing and shuffling process. It's not a bifurcating process. It's like taking two leaves of the tree and chopping them up and mixing them back together. So what happens is that if you apply tree methods to var genes, you get what I'm calling garbage, because the model and data are mismatched. So this tree, which is like a typical tree but arranged around a circle, doesn't really tell us anything. All of these genes around the outside are just connected to the center, which is to say, we have no good explanation, so here's your diagram. So understanding these var genes means that we need better methods. So what we're going to do here is develop a network-based method. We're going to focus on the DBL alpha domain, which is present in almost all var genes. So that's this blue domain here. A tip to the graduate students in the room, it's really important to choose a project that you really feel a connection with. And here, working on the Duffy binding-like domains, really nice because DBL are also my initials, so I feel a real connection to this problem. But anyway, we actually analyze this because this domain shows up in almost all the var genes. Because the protein needs to still bind to the endothelial cells, right? It can't lose that function. We see some structure that's preserved. So the signal is in the variable regions outside of the conserved regions of this. So what I'm showing you here is some rough measure of what we call conservation. So if I compare one gene to all the others, what I see is there are some regions where it's similar to all the others, similar, 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 and then in between, it's just one gene is totally different from all the rest. So these highly variable regions where we're alternating variable, conserved, allows the parasite to have these genes that can still bind, but are as random as possible. So we're going to analyze these highly variable regions. And the overall picture looks like this. If I can get genetic sequences from different parasites, we're going to avoid looking at those conserved regions because we know what's happening there. We're getting protein structure. What we want to understand is how the parasite evades immunity. And so we're going to look in the variable regions by basically covering up the conserved regions in between, and then each one of these will build a network. We're going to build separate networks at each one of these because what we don't want to do is constrain our analysis ahead of time to assume that the evolution at every one of these parts of the protein will be the same. If one part of the protein kind of sticks out one way and another part sticks out another way, the selective pressures on those different pieces of the protein could be very different. So what do we actually do? At each highly variable region, we're going to map the sequences to an all-to-all weighted network. So here are 3 actual amino acid sequences within a highly variable region. For each sequence, I write down a node represented by a circle. And now I'm going to choose the weight between those nodes to be the integer length k of the longest shared k-mer or substring. So for the top and the bottom, you can see this block here is of length 16. So I connect the top and the bottom with an edge of weight 16. Fine. A long shared substring implies a recent recombination or some kind of hereditary relationship between these genes. They're related somehow. So what we're going to do is build this all-to-all weighted network and then do something that you're not supposed to do, which is we're going to threshold it and say, if the links are below some threshold, then we delete them. If they're above the threshold, then we just keep them and treat our data as binary. We have to do this because we don't know how to interpret the lengths. It's really an active decision. The biological difference between some length 8 shared substring and a length 16 shared substring, we don't know if this is twice as meaningful as this, because we don't have a sense of scale here. So instead, we're just going to say above the line counts as related, below the line doesn't. And then do the typical thing in data analysis where we vary that threshold and then see if it makes a difference. Okay. The final step in this procedure is going to be to find community structure in the networks. So if functional or evolutionary constraints are shaping which genes get to recombine with which others, then the network will have community structure. We'll have these genes that tend to recombine with each other, these other genes that tend to recombine with each other. But if there's not much mixing in between, then we'll have fewer links in between those communities. So if we build the networks and then use a way to find structure, then we can start to analyze, there's some kind of mechanism in the body or in the binding process that's actually pushing these genes to be separate from the adjacent groups. So that's the overview of the idea in theory. But as it turns out, network visualization can really help us out here because it shows us that we're on the wrong track. If I visualize one of these highly variable region networks, I see a problem, which is that there are lots of all-to-all clusters or cliques embedded in this network. So the reason that I think this is a problem is because cliques are very low probability under most community detection models. So if you ask yourself, if I were given a network with no labels on it, how would I find community structure? You need some kind of mathematical model or some kind of Hamiltonian that you can just maximize over your system in order to find community structure, to partition the network into groups. But having those cliques in there really makes that a big struggle. So one popular method called modularity struggles when you have cliques of different sizes and it hits something called the resolution limit. And it struggles especially when cliques are embedded in larger networks, which is exactly the case that we have here. There are some nice statistically principled methods based on random graphs. These are stochastic block models. But they create cliques in their generative form with probability 0, which means when we flip things and try to infer community structure, we really struggle. The reason here is that cliques are formed with very low probability in random graphs. And as the clique gets bigger, that probability goes to 0 geometrically fast. So rather than trying to apply models that don't match our data again, we're instead going to ask, why are there cliques in the first place? And I know what you're all thinking. It's Kevin Bacon. So, has anybody played this game, 6 Degrees of Kevin Bacon? Where you like name an actor, and then you have to name somebody that co-starred with that actor. And it's really an argument about the diameter of co-starring networks, which is that within 6 hops you can get to Kevin Bacon. Or within K hops you can get to Erdos, right, if you're thinking about Erdos numbers. Okay, so what is 6 Degrees of Kevin Bacon? I can think about Bono here. Bono was in the $1 Million Hotel. So was Charlene Woodward. Charlene Woodward was in He Said, She Said, and Kevin Bacon was also in He Said, She Said. So really, it's 2 hops to get from Bono to Charlene Woodward to Kevin Bacon. What we're actually doing here is something different if we zoom out just a little bit more. This is a bipartite network. We have actors who are in movies that hire actors. So bipartite network is a network in which there are 2 different types of nodes, and the rule is you only get to have links between the 2 different types of nodes. So if you want, you can think about this as a vertex coloring problem, where the graph can be colored by just 2 colors. So here's where the problem is. In addition to Bono being in the $1 Million Hotel, Mia Jovovich and Mel Gibson were also in that, as well as a bunch of other people. So if I were to go back to the co-starring network, the fact that all of these people were in the same movie means that when I project the bipartite network into a co-starring network, Bono connects to the rest of the cast, Mia connects to the rest of the cast, Mel to the rest of the cast, and so on. In other words, taking a bipartite network and projecting it just down to a co-starring network creates cliques. And that's exactly the problem that we have here. So it turns out there are lots of different networks that are what we call one-mode projections of bipartite graphs. So here we're thinking about genes and their constituent substrates. And in fact, if I looked at the diagram for long enough, we would have seen this, that we're explicitly calling out the substring that connects the 2 genes in these different cases. But we just throw it away when we write down the projection. So we have lots of examples of these, plants and pollinators, papers and authors, documents and words. social network users and the locations where they access. So all co-authorship or co-starring or co-whatever networks are really implicitly one-mode projections of these larger networks. So when somebody asks you, oh, do you know what your Erdos number is? What you're really doing is a shortest path calculation on the projection of a larger bipartite network, which is authors to papers. And you're asking how many 2 hops in the bipartite network does it take to get to Erdos? Okay. So we've avoided model data mismatch because instead of looking at these unipartite projections where we have the cliques, now we're saying, okay, these are really bipartite networks. But now we have a new problem, which means we need to be able to find community structure in bipartite networks. Now, just briefly to talk about the premise of community structure, usually what we're thinking of is something like clustering. We want to find a set of nodes that are somehow similar to each other. But in a bipartite network, the nodes that are similar to each other aren't connected to each other because the rule is you can only connect across the network to the other side. So we need a different method. So here's where we get to do a little bit of math. Our goal is going to be to find communities in a bipartite network. So we can ask, what are communities in a bipartite network? Like, what would it mean for there to be communities? So of course, if you like generative models, if you want to find something in data, you ask, how can I generate it? So that's what we're going to do. So let's imagine that I have my 2 types of nodes. I'll call them 1 and 2. I'm going to imagine that I just specify N1 types 1 nodes and N2 type 2 nodes. If I want to generate community structure, I better divide these into groups. So that's what I'll do next. So now I'm going to say that there are B1 blocks of type 1 and B2 blocks of type 2. I'm just going to say that BI is the block assignment of node I for some bookkeeping. I want to specify a little bit more because some of these nodes are going to have a propensity to really show up in a lot of sequences or have a lot of different k-mers in them. So I'm going to specify the degree of node I. So let's say I specify the degrees of all these. The degree is just a number of connections. So if these are sequences, 3 would be the number of k-mers that this sequence is going to connect. Next, I want there to be block structure or community structure. So I'm going to specify this matrix, ERS, that counts the number of edges between groups R and S. So in this case, here's my block edge matrix that says light yellow to orange, I'm going to put 4 connections. Light yellow to dark red, I'm going to put 0 connections. I expect 0 from here to here. Now we have some constraint satisfaction problem. Can I write down the edges in a way that matches the constraints of my generative model? And up to those constraints, I'm just going to do everything else uniformly at random. So that's what we do. So here's one possible configuration. So this network configuration matches my degrees vector, my block structure vector, and my group membership assignments. Cool. So that's one option, but of course, there are lots of different options. So for the same constraints, this was our original example. But if I swap the 2 blue edges, I get this example. Notice that I still have the same block-to-block connectivity. I still have the same degrees on all the nodes. If I then swap the green edge with one of the blue edges, I get this network. And again, this one also satisfies. So you can imagine that now we kind of have some sort of like combinatorics problem. How many ways are there of arranging these edges in a network where I'm specifying the degrees and I'm specifying the number of connections from one block to another block? But this is what it would mean to generate block structure in a bipartite network. Okay. So another way of saying this is that a choice of parameters gives us an ensemble of networks. And the size of that ensemble can be 0 if our sequence is non-graphical, for instance. There's no network that actually satisfies the constraints. It could be size 1 if there's only one network that satisfies the constraints, or it could be combinatorially large if we have a huge variety in the networks that we could produce given some set of constraints. So this is what we call the micro-canonical ensemble of the stochastic block model. We're going to represent it by omega KEV. Omega just corresponds to this set of possible networks that meet those constraints. Cool. So we like generative models because they open the door to inference. And what I mean is that if I give you KEV, you can draw stochastically somehow and generate network data with an adjacent matrix of A, where A just represents which nodes are connected to which nodes. But similarly, if you have network data A, we can perform inference to find the parameters K and B. And the key here is that when we infer the assignment of nodes to blocks, that's this vector B, we found the communities that we seek. We found a partition of the nodes into groups. So inference of the microcanonical stochastic block model is equivalent to community detection. And we happen to get the edge connectivity matrix and the degrees as well. So if I'm thinking about this ensemble as just a set of networks, such that the probability of choosing any network from that set is equal, and the probability of drawing any specific graph from that ensemble is the likelihood of the data given the model parameters, and that's just 1 over the size of that ensemble. So the upshot of this is that if you can compute the size of this ensemble, if you're really good at counting things, in other words, then you already have the likelihood function. So the problem here is really overfitting. When I say overfitting, what I mean is, what if I just let the number of groups be the number of nodes, so every node is now in its own group? Then the group-to-group connection matrix should just be the node-to-node connection matrix. But I haven't modeled the data here, I've memorized it. The model has basically memorized the data, which means that, yes, I will have maximized my likelihood, because this ensemble will be really small. It'll have A and maybe a few other variants in it. But I haven't really learned anything. I haven't divided the network into meaningful groups. I've just said that every node is its own group. What we need is some kind of complexity control, some way of telling the model that we want a parsimonious view of the data. Yes, compress it. Yes, model it. Not too much, not too little. So here's what we do. If I look at the full joint distribution, I can always factor it into likelihood and then a prior. And here we're looking at this nice Bayesian hierarchy where I'm breaking down the full prior into different pieces. If I take logs and I stop factorizing the prior, I can see this is a log of likelihood and a log of the prior. If I plug in what I know about the likelihood itself, that's just the size of omega to the -1. It's a probability we had on the previous slide. And of course, using logarithms that we teach the undergrads, the minus can come down. I have minus log of the size of the ensemble. Of course, the entropy of a uniform distribution with k states is minus log k. So when I see this, I'm thinking that this is really the entropy S of this ensemble omega. And the entropy here has a really nice interpretation if I'm thinking about this in terms of bits, or if we're using natural logs in nats. This is like saying, how many bits does it take to explain the data given the model? In other words, how well does the model actually compress the data? So if we're in the case where the model just memorizes the data, then it achieves almost perfect compression. And if it totally memorizes the data, it's perfect compression. The model is the data. But there are some number of bits that I require to explain the model. And if I add these up, I get something called the description length, DL, which you'll notice is, again, my initials. So again, I feel another connection. But the idea here is that we're going to try to minimize the description length. This asks for a parsimonious description of the data and the model. And asymptotically, this is equivalent to using BIC, or the Bayesian Information Criterion. So here's a better explanation of minimum description length. Let's say that I have some message, and I want to transmit it to somebody else. Now, I could transmit that message in its full beauty. All the words and spaces are there, and maybe some diagrams too. And it takes some amount to just transmit the information in its raw form. But I could come up with some kind of compression scheme, maybe where I collapse certain phrases together and I just represent those by a symbol. And so now what I transmit is the compressed message. But if you want to decompress it on the other end, you need the codebook. So I have to send you the compressed message and the codebook. If I go all the way to the other end, I send you a message of just 1 bit. It says, look at the codebook. And then the codebook itself is the message. So in this extreme case that we've gotten to, the codebook has memorized the data, the model is too big. but the entropy required to compress the data given the model is really small. On the other end, I'm just sending you the raw data and not using any model at all. And you can prove that there exists a minimum in between, where you've compressed the data and you have a reasonably sized model, and so you have a minimum in this description length. So that's what we do. Minimize the description length, and it's sort of like a statistical version of Occam's razor. We want the simplest description of the data that I can get. Okay, so what we did is we wrote down all the counting needed to actually minimize the description length for bipartite networks. And then we set up a quick test to see whether or not it mattered. Like, did writing this down for bipartite networks actually make a difference? And do bipartite priors and bipartite counting do any better than sort of the typical kind of counting? So we had two tests. One is an easy test and one is a hard test. The idea is, in the block matrix, so these are nodes here, and each one of these darker areas is a block of high density where you have a group on one side that connects a lot to a group on the other side. And what we do is we mix in a bunch of noise. And we mix in increasing amounts of noise, and as the noise level goes up, the pure block structure gets obscured by the noise and we have trouble recovering it. And so the question is, how much noise are we able to tolerate before the method totally fails? So what you can see here is how well we're doing in recovering the known answer that we planted in the data. As you can see, as I increase the amount of noise, my ability to recover goes down and down and down. In a hard test, where the structure isn't so simple, now it's more complicated. I have a different number of groups for one side of the network versus the other. You can see that the same general trend is clear, that as I add more noise, it gets harder and harder. What I want to point out here is that if I compare the dark blue with the medium blue here, the circles and the diamonds, you can see the difference between the bipartite math and two-dimensional search versus the unipartite math and two-dimensional search. So we're holding fixed the search algorithm and just changing the math, and you can see that there's not that much difference in the easy case, but we do see a difference when the going gets tough. So here, the bipartite math is better able to recover this complicated structure than the unipartite math, which collapses down to just basically guessing more quickly. We can also control the other way. We can compare the unipartite math and my graduate student Suchi Yen's algorithm to search the space versus the unipartite math and the unipartite search algorithm. So that's comparing these diamonds and these triangles, and you can see a difference between them here and another difference between them here. So our conclusion, just from this cheap synthetic example, is that the bipartite math, when you're writing down the number of configurations in these ensembles, really matters. The priors really matter, and the search matters. So instead of searching in this one-dimensional space, where you're really changing the number of communities, what we're doing here is searching over a two-dimensional space. I'm asking how many communities are there on one side of the network, how many communities are there on the other side of the network. Of course, the algorithm needs to be more complicated because now we're trying to find sort of a peak in the two-dimensional space instead of a valley in that two-dimensional space instead of a single valley in a one-dimensional space. But the two-dimensional search turns out to do much better.
How do you ensure that 0.39 line that it will fall amount?
Yeah, so what's going on here? This quantity is called adjusted mutual information. It's sort of like mutual information normalized by the entropies of the original distributions, and then you account for like how much overlap do you expect between two partitions just by chance. So what's happening here is that the partition that all of these methods is returning is just, well, I don't really see any structure here because it's all random. And so it just says one community on this side and one community on that side. And the amount of adjusted mutual information between that partition, one group and one group, and our true partition, this here, is that value.
I see. So that's really a consequence of your specific setup here.
Yeah.
Yeah, that's very specific.
Yeah, so the baseline here is just guessing, like, I don't know anything, so it's just, the network can be divided in half. Okay. So the whole point of this tangent was that we can avoid phylogenetic trees and instead use networks. But the networks that are unipartite projections have cliques, so that's no good, so we're going to use bipartite networks. If we want to use bipartite networks, then we need bipartite community detection methods. And so that's the point where we are now. Let's go back to malaria. Unfortunately, when I was analyzing the malaria genes, I didn't get to work with Suu Kyi Yin, and we weren't preparing this paper that uses these nice methods. So instead, what I'll show you is using the methods that I came up with as a postdoc, which were also stochastic block models, but where we fix the number of communities and avoid this minimum description method. We hadn't solved that problem yet, so there's a disclaimer here that we're using older methods. But anyway, back to malaria. We took data from 7 parasite genomes from around the globe, and that was 307 different VAR sequences. We found 9 highly variable regions, so we built 9 networks. We used each highly variable region's length and amino acid composition to select the substring length cutoff. Basically, how small are the substrings below which we don't care. Then we identified communities separately for each highly variable region using the old method. And we found that only 6 out of the 9 highly variable regions exhibited non-random recombination patterns. In other words, 3 out of the 9 were basically random. We didn't see strong community structure. So this is a picture of what things look like coming out of the parametric model, where we've asked for 3 communities on one side, 3 communities on the other side. The big circles here represent genes. The smaller circles represent substrings. And so if you look closely, you'll notice that the genes connect to substrings, connect to genes, but no gene-to-gene connections. So what we can ask is the non-random structure consistent across the highly variable regions? So if the evolutionary constraints are the same at 2 of the highly variable regions, we expect a similar large-scale structure, because they're both being constrained by the same rules. If you have groups on one side, you should have groups in the other location too. On the other hand, if there are evolutionary constraints that are different at 2 highly variable regions, we expect different large-scale structures. So yeah, we see community structure on one highly variable region, and yeah, we see it at the next and the next. But those structures are different from each other. And finally, if the evolution is unconstrained at a highly variable region, we expect weak or basically no structure. We expect that network to be better modeled by a random graph than something with blocks. What we're going to do is compute pairwise distances between the highly variable regions communities using this thing called variation of information, where I just take the partitions into different groups, treat them like random variables, and I compute the entropy of one, entropy of the other, subtract off 2 times the mutual information. What we like about this is that this is a proper metric. So when we're doing lots of pairwise comparisons, we know that we get the triangle inequality on our side. We feel better about that. So here's what things look like. I'm showing you the 6 highly variable regions, 1, 5, 6, 7, 8, 9, and the distance in variation of information to the other highly variable regions. So what we see is that 4 of these highly variable regions exhibited different non-random evolutionary constraints, so that's 5, 7, 8, and 9, that are typically far from each other and far from the other HBRs. Meanwhile, HBRs 1 and 6 exhibited similar constraints, distance to each other, 1 to 6 and 6 to 1, was a lot smaller. So what this suggests is that 6 and 1 are somehow coupled in their evolution, whereas the others are evolving independently. So where we ended up here is with a hypothesis for how this gene family actually evolves. These highly variable regions enable a parallel and partially independent VAR evolution. So most of these highly variable regions are structured, so they're non-random. They appear to evolve under independent constraints. So those are these red stripes here. But some regions, like 1 and 6, exhibit similar communities, implying that their evolutionary is coupled somehow through similar or coordinated constraints. So why would you have coordinated constraints? Well, maybe you need coordinated function like endothelial adhesion or protein structure. That if you don't have these 2 pieces of protein moving together, then it no longer folds correctly or no longer binds correctly. On the other hand, once you've satisfied these coordinated constraints, you really want, if you're a parasite, as much diversity as possible to evade the immune system. And so you can do that by making these evolve independently. So that's kind of the picture that we get to when we use these networks, community detection methods, and compare the communities that we find to see how these genes are structured as a gene family. Okay. So, what can we say about the VAR population structure in Senegal? Let's find out. The parasite population has been adapting in this place called Chess, Senegal. If you've ever been to Dakar, which is right on the coast, Chess is about an hour or 2 hours inland. So the relative incidence of malaria relative to 2006 is going down, and then it's starting to kick back up. Okay, not my regression model. But anyway, if you do ocular regression, yeah, it's going back up. Fine. So this is a malaria rebound in Chess, where malaria is at a pretty low prevalence. The public health infrastructure there is better. The problem is that the VAR-specific antibody responses, these little antibody guys here, wane in the absence of exposure. So you have an interesting sort of dynamical system here, which is that stimulation from the parasites increases the population level of immunity, but when the parasites go away, people's immune systems forget. So if a parasite is then reintroduced, people will be more prone to getting very sick. So we can think that time and exposure basically modify the selection landscape of the population. Because again, these are the antibodies that are pushing the VAR genes around to evolve. So you have this nice two-way coupled dynamical system. So what we see is some evidence of time-dependent population recognition of VAR genes in this area. So what I've done here is taken the gray dots here represent those 7 genomes that we analyzed before. This is a unipartite projection now. And then taken 2 2009 parasites, that's red and blue, with matching genetic barcodes. And then what we find is that these have similar VAR repertoires. So the red and the blue here tend to show up near each other throughout this network. The yellow is a control from the previous year. So if I actually look at the VAR genes that we get from sequencing here, and I say that 2 VAR genes I'm going to connect in this little diagram if they're like 95% similar or above. What I can see is that the 2 parasites we collected in 2009 have a lot in their intersection, whereas the 2008 control didn't overlap very much. The key here is what we can do with immunity. What we can ask is, among the plasma samples that we collect from people in the population, what fraction of those plasma samples recognize these parasite antigens? We just do this assay and ask what fraction respond. So if I take 2008 plasmas and I ask them about the 2009 parasite, they don't recognize. The 2008 parasite, or the plasmids, also don't recognize the 2009 parasite. It's a parasite from the future. They haven't learned it yet. 2008 control is well recognized by the 2008 plasmids. Now we compare with the 2009 plasmids, and we see that 2009 immune systems have learned the 2009 parasite, and they also know the control. So the suggestion here is that the immune systems in the population are learning how to respond to these genes. So we have some evidence that the immune systems are actually pushing back on the evolution of the genes. Okay. So we want to expand this study. What we do is over 2007 to 2014, collect lots of different what we call haplotypes, where these are parasites that have an identical genetic barcode. All it means is that we look at 24 locations that are under neutral selection for SNPs, and we find that we can segregate the different parasites into these groups. But once we have these groupings, we can see what happens to prevalence according to the groupings over the years. And I want to look at this dark maroon color. So here we see that haplotype 29 seems to adapt to the population using a VAR repertoire that's unknown. So it gets a little foothold here and expands in prevalence because the immune systems of the population don't know the parasite's tricks yet. In this year, 2011, the parasite in haplotype 29 accounts for 25% of the cases, but then the immune systems start to learn, and they start to catch up. And they push that parasite out, so the human population's immune landscape has adapted at this point, clearing haplotype 29. Cool. So it's a cute story, but we want to know whether or not the data actually support this story. So we can ask, do the clusters that are formed by these barcodes have identical VAR repertoires? We're putting things into groups based on things that are not immune evasion genes, and assuming that the immune evasion genes are the same. So we need a test to figure out if that's the case. So we have a special challenge when we compare 2 parasites' VAR repertoires. which is that because there's 60 bars per genome, turns out they're accessible only by this technique called PCR, which is slow, expensive, and stochastic. Because the genes are so recombinant and shuffled, the way that we typically assemble genes from short reads is by using the fact that they partially overlap to stitch together a nice assembly of the whole genome. But if you have a bunch of genes whose structure looks like lots of little pieces that are shredded and overlapped, then you can't assemble the genes. So we have to use this thing called PCR. So the question that we'll now address is how should we estimate repertoire overlap from samples of our genes? So here is another little math problem. How should we compute bar repertoire overlap? We'll set it up in the following way. I have repertoire A, and let's imagine that I have 60 dots here. I have repertoire B, another 60 dots. And I have some that are in the intersection. I have repertoire overlap. I take some sample from A, some sample from B, and what I see in my observation is some number of parasite genes that are in the overlap of the sample in some size of the sample from each. The problem is, I do this experiment again, and I get a different observation. In this observation, there's no overlap. In this observation, there's 50% overlap. In my actual data, there's neither 0 nor 50% overlap. So because this process is stochastic, repeated observations are going to be inconsistent. Seems pretty straightforward. We can think about this the other way too. Let's say that we have two different realities. Here's one reality, here's another reality, and just by sampling, we end up with the same amount of overlap in the sample. Fine. So the question is, how should we estimate the true overlap, knowing that our single observation is noisy? So we do a lit review, right? Okay, so it's kind of fun. Where else does this problem come up? It turns out that this is a core problem in ecology, which is I want to compare sets of species. So does anyone know what this is? See your lawn?
Quadrat?
It's a quadrat. Yeah. So that's a quadrat with flowers. Here's a quadrat with person. Here's one with a GoPro. So the technique here is I want to understand sort of relative overlap between populations. So I take my quadrat, throw it in the air, wherever it lands. I document what I find inside the box. I do it somewhere else, and I document what I find inside the box, and then I can compare it. So the shared diversity between two populations, the ecologist called beta diversity. And it goes back to Torvald Sorensen, who's a botanist. And he said, okay, well, if you have NA samples in one quadrat and NB in another, and you have NAB shared between them, and you just take the size of the intersection and you divide it by the average of your samples. Intuitive? If you have identical populations, you're going to get 1. If they're unrelated, you're going to get 0, because it has some nice properties. Cons. No notion of uncertainty. It assumes we have complete sampling. We have a downward bias whose strength depends on the sampling rates. So to give you an idea, I want you to consider the difference between these two scenarios. In one case, I get 40 samples from each population, and I see nothing in the intersection. In another scenario, I get only 10 samples from each population, and nothing in the intersection. While the estimate for both of these would be 0, I have a lot more certainty in the case where I have 40 samples than in the case where I have 10 samples. Another example, imagine I had 2 sets of 10 objects each with 5 objects in the intersection. In principle, I should have a 0.5 overlap. But if we repeatedly just sample 9 out of the 10 objects from each set, and then compare and compute this overlap, we average 0.45. So we're off by 10% just because we only had 90% sampling rate instead of 100%. So it turns out there are 20 different algebraic combinations of NA, NB, and NAB. All of them inherit these issues caused by subsampling, include another famous botanist named Jacquard, whose way of combining NA, NB, and NAB is a distance metric, but also suffers from this problem. So the key realization here is that this is a random variable. We just have to treat it as such. So let's model the sampling problem in the forward direction. Same trick as before. If we can figure out how we generate the data, then we can reverse it and do inference. So if the reality is that we have 60, 60, and some overlap with S, and I want to know what's the likelihood of observing NAB in the intersection given NA and NB, let's fix S, NA and NB, write down the likelihood. So if I started out with 60 objects, S of which were special, and I subsample, then I'm drawing a sample of NA objects from the 60, and the number of special objects I get is going to be SA. And if the probabilities are uniform and I don't replace, it's a hypergeometric. So now I think about things from Green's point of view. Green is going to sample NB, but its number of special objects that it could grab from is no longer S, it's SA. So NAB ends up being a hypergeometric where there are SA, special objects, 60 total, and sampling. So I have this nested hypergeometric process, which means that I can write down the probability or the likelihood of observing NAB in the intersection given a true intersection in some sample sizes. Of course, the way I'll compute that is to sum over this intermediate variable for all its possible values for these two conditional statements. So we can use this likelihood of the uniform prior to compute a Bayesian estimate of our original overlap. What that looks like in practice is we can get some posterior distribution over the amount of overlap. So here's the overlap between 0 and 60. Here's our posterior distribution. And what's nice is that we get an estimator of the overlap via the mean of the posterior. So here's the expectation of this distribution. We can get a credible interval to quantify the uncertainty in our estimate. And we have Bayes optimality, so this approach minimizes the posterior expected loss, assuming that we have the right data generating mechanism. Notice that our Sorensen-Dice coefficient comes in right here. So it's providing a much smaller estimate than our Bayesian estimate. Okay. So we can test statistical consistency. I'll just sort of skip through this quickly and just say that by varying the rate at which we subsample, so terrible subsampling, bad, not so bad, you can see that all the estimates converge on the true value, but the Sorensen-Dice coefficient, which are the x's, converge from below. So there's a bias here. The bias depends on how bad the subsampling is. What we then did was we went back to the literature and said, if we know that the rate of subsampling affects how bad the bias is, what about these comparative studies where people are looking at data sets next to each other where you have different rates of subsampling? The biases in these are going to be different, so if we can correct for the biases, we may learn something different about the past. So here's us using the Sorensen method. These are clones from Arikemes, Brazil, Amale, Papua New Guinea, and Kalihi, which is in Kenya. And this is the reanalysis. So what I want to point out is that under reanalysis, these clones, which we know are clones and they should be identical, go from looking like they're about 30 out of 60 similar to looking much more like what they should be, around 60 out of 60 similar. Similarly, these clones from, or the parasites from Papua New Guinea go from looking like they're very dissimilar to looking like they're actually quite similar. So using the different methods really changes the conclusions, but in a way that varies depending on how much data these different studies had. But as it turns out, we shouldn't stop here because we shouldn't be interpreting these data sets at all. So if I ask, what's the width of the credible interval in my posterior estimates? How certain am I about these different estimates? If I look at the clones, what I see is that the credible interval is typically between 0 and 30. So what that means is that if I estimate 40, that's like 40 plus or minus 20. It's uncertain, but it's not nearly as bad as the estimates from Papua New Guinea. So these estimates here are like saying these two parasites overlap with 30 of their repertoire genes, plus or minus 30. So the amount of uncertainty here is on the same order of magnitude as the estimates themselves, which suggests, actually in the analyses, they shouldn't even be here. This is a study that needed more sampling. You need about twice as many resources to even be worthwhile scientifically. So what's nice is that now we can plan studies with the future uncertainty of our estimates in mind. which means that we know how to write a budget for the next NIH proposal. So you choose your uncertainty, and then the math can tell you what your sampling depth ought to be. So that's nice. Okay. I want to go back to Senegal. I'm going to move quickly here because I know that we're just about out of time. But what we can now do is compute these repertoire overlap networks for a few different haplotypes, where each node is a parasite isolate. The edges are weighted by our now Bayesian repertoire overlap estimates. So these are ex vivo var transcription. It's not important exactly what that is, but the clusters that we see in this network correspond exactly to the haplotypes that we see here. So that suggests that the genetic barcode is the same as the var repertoires, but still with some amount of overlap between them. So we can look at the genomic var repertoires, we can look at var transcription, we can look at culture-adapted var transcription, but the punchline is similar barcode means similar var genes and similar expression. Cool. Skip that. So in conclusion, what I talked about today is that we can write down these nice network methods to show how Plasmodium falciparum var genes evolved to balance antigenic novelty with maintenance of binding phenotypes. In other words, you want as much variability as possible, but not so much that it breaks your protein. Talked a little bit about inference methods that let us rigorously compare parasites when the underlying data are really flawed, pointing to a need, I think, for a reassessment of past results now that we have better methods. Finally, when we apply these methods, it really opens the door to a var genomic epi, some exciting initial results in a low transmission setting in Senegal. So what's next? Eric Johnson is rebuilding the analysis to look at other var gene domains in addition to DBL alpha. Suchi Yen is developing those better and faster community detection algorithms. Suchi and Kate are trying to see if pairwise overlap can be done directly from short reads, so we can unlock loads of unassembled var whole gene sequencing data. So this idea is really cool. Don't stitch the genes together in the first place, just compare the piles of unassembled reads to each other, and then figure out how much the repertoires have in common. Sidestep another difficult process. And then my primary collaborator in all this work, Amy Bay, who's at Yale School of Public Health, she and I are now turning to the immunity side of this dynamic system to understand how the humans are shaping the parasites, how the parasites are shaping the humans. So I'll wrap up here. I want to thank Kate, Eric, Suchi, and Suchi. It's a little confusing, as well as my collaborators at Yale, Harvard, and Colorado. Thanks. Love to answer any questions.
Thanks for a fun talk. Any questions?
Yeah.
This may not be possible just because of the length of your study, but have you seen, you were talking about how a low transmission setting, like the immunity system unlearns, right? Have you seen that actually happen? Was that why HAP29 was something you were interested in? Was that one that was present before they learned it and they unlearned it? Or is that something we don't see on these timescales yet?
So a weird thing that we see when the transmission gets low is that you kind of have it's almost like a trending haplotype. A certain parasite becomes popular and gets very prevalent in the population. The immune system seems to learn it and then push it out again. When transmission is higher, you don't see that. And I think what's happening is that the people who have that wannabe trending haplotype are also getting what's called superinfected. So they get bitten by another mosquito, and now they have two different parasites in them at once. When those two parasites get sucked up by the next mosquito, they do what's called genetic reassortment. So it's like when everybody's parents made us, right? You get some chromosomes from one parent, some chromosomes from the other, so you have a much clearer mixing process. So anytime you would have one kind of trending parasite that would want to become popular, it gets mixed out.
And that's in a high.
That's in a high transmission setting. You get that
super infection.
Yeah, great. Any other questions?
It's surprising that the immune system can adapt so quickly. To me, it's surprising that it does. I don't know anything about it. You said like from 1 year to the next, you can see that the system is already evolving.
Yeah, I mean, so we see the same kind of adaptation with things like influenza, right, which is why we need a different influenza vaccine each year, and why they're trying to predict kind of where the virus is going to go that year, so they can get the vaccine in front of it. To me, the more remarkable thing is actually the other way. So chickenpox, one antigen, the vaccine targets that antigen, you're fine. You've learned that lesson for like polio. There are like 3 main serotypes, one of which was eliminated like 2 weeks ago. But if you make a trivalent vaccine that hits all 3 of those, then nobody gets polio. But in this case, for a single parasite, you need 60 variants to teach the immune system that many lessons. And then you just find some other parasite that has another 60. And so it's just sort of hopeless to make a vaccine. But I actually have one small bit of hope, which is that if we can see some of this community structure among the VAR genes, we can identify among those VAR genes which are the ones that, when highly expressed, lead to more severe malaria symptoms. We just make a vaccine against that. And it doesn't prevent disease, but it does prevent severe disease. So if we turn malaria from something that kills people into something that makes them sick, like that would be a really good. There's also hope for targeting one particular VAR gene that only binds in the placenta of pregnant women, which means it's doubly bad because it threatens the baby as well. If you could develop a vaccine for that, then I think that would also be good.
Last chance, any other questions?
Thanks.
Thank you.